Welcome to Rad on Smart Review, RadBio edition.
Today, we're diving into something really fundamental, a concept that truly impacts how we deliver radiation and predict its effects: dose rate.
That's right. It sounds simple, maybe, but the speed of delivery matters. We'll cover the general rule slower, often means less cell kill, but also a surprising exception called the inverse dose rate effect.
And we have to talk about LET, linear energy transfer, because that changes everything about the dose rate effect.
Absolutely. We'll also, touch on why this is also critical in the clinic. Think brachytherapy, TBI, even understanding radiation exposure should be a good discussion, maybe even some board tips along the way.
Definitely. Let's start with the basics.
Okay, foundation one, the general dose rate effect. For low LET radiation, our standard X-rays, gamma rays, the basic principle is deliver the dose slower, you generally get less cell killing.
Compared to zapping it all at once, yeah. And the core reason, the absolute bedrock here, is repair of sublethal damage. Cells are constantly trying to fix themselves.
Exactly. So if you hit them with a high dose rate, you're causing damage really fast.
Right.
It can just overwhelm those repair crews.
Right. Or you get multiple smaller hits, sublethal lesions, happening so close together in time they interact and become one big lethal hit before the cell can fix them.
But slow it down. Low dose rate.
Now you're giving the cell a fighting chance. The damage happens more gradually, those repair mechanisms. They're working during the radiation exposure.
So they can't just patch things up before it's too late.
Yeah, they can fix a lot of that sublethal damage before it accumulates or combines into something irreparable. It's a continuous process.
And this changes the survival curve, right?
Yeah.
That classic graph.
It absolutely does. As you decrease the dose rate, the shoulder of the curve representing that capacity for sublethal damage repair, it gets smaller. And the overall slope gets shallower.
Meaning you need more total dose to kill the same fraction of cells.
Precisely. And if you go to really, really low dose rates, the curve can look almost like a straight line, almost exponential.
Why is that?
Because repair is keeping up so well with the sublethal damage, most of the killing that happens is from those single-hit irreparable events. That's mainly the alpha component in the linear quadratic model dominating.
Okay, so dose rate matters, but you mentioned ranges. It's not like every little change has a huge effect.
Right, it matters most in a specific window. Think about the extremes first. Very high dose rate, say over 1 gray per minute or 60 grays per hour, the radiation is delivered so quickly.
The whole thing's over before the cell even knows what hit it, repair-wise.
Pretty much. There's virtually no time for repair during the irradiation itself. The effect is basically the same as an instantaneous acute hit. Whether it's 1 Jimin or 10 Jimin, the immediate biological punch is similar.
And the other end, super slow.
Yeah, very low dose rates, less than, say, 0.01 gray per minute or 0.6 gray per hour. Here, the damage is trickling in so slowly.
Repair keeps up easily.
Exactly. Repair mechanisms handle it efficiently. And if the exposure goes on long enough, something else happens. Cell proliferation. Repopulation.
So the cells can actually divide and replace the ones that are killed or damaged.
Yep. Repopulation can start to offset the cell killing, especially over days or weeks.
So where's the action then? Where does dose rate really make the biggest difference?
It's that intermediate range, roughly between, let's say, 0.01 borough per minute and 1 borough per minute, or 0.6 to 60 borough per hour.
Why there?
Because that's where the balance is most dynamic. The rate of damage induction, the capacity for repair, and how cells are progressing through the cell cycle, they're all interacting intensely. Small changes in dose rate here can really shift that balance and significantly alter the biological outcome.
Okay, that makes sense. Now, about that exception you mentioned, the inverse dose rate effect.
Yes. This one's a bit of a classic, especially for exams. It's completely counterintuitive. Here, for certain cell lines, decreasing the dose rate actually increases cell killing.
Wait, slower means more killing? How does that work?
It feels wrong, doesn't it? It's seen in specific cell lines. HeLa cells are the famous example, but only within a very specific, narrow range of low dose rates, maybe around 0.3 to 0.7 gray per hour for HeLa, something like that.
So what's the mechanism? It can't be about repair time then.
No, it's about the cell cycle. The accepted explanation is cell cycle redistribution, leading to a G2 block.
G2 block. Okay, I'll unpack that.
At these specific low dose rates, the radiation isn't hitting hard enough to slam the brakes on the cell cycle early, like in G1 or S phase. So cells keep progressing.
They keep moving along.
But the continuous low-level damage is just enough to trigger a checkpoint delay, a block, specifically in the G2 phase of the cell cycle, right before mitosis.
In G2, that's a sensitive phase, isn't it?
Highly radiosensitive. G2 and M phase are the most vulnerable points. So what happens is during this prolonged low dose rate exposure, cells tend to pile up, accumulate in this highly sensitive G2 phase.
So you're essentially enriching the population with the cells that are easiest to kill, right when they're being irradiated.
Exactly. So compared to a slightly higher dose rate where maybe they arrest earlier or repair better, or a much lower dose rate where they might slip through the G2 block or repopulation kicks in, this specific intermediate low rate traps them in G2M, leading to more overall killing.
Wow. Okay, that is counterintuitive, but makes sense with the G2 unsensitivity. And you said this pops up on boards.
Definitely. Knowing the inverse dose rate effect and its link to the G2M block is high yield. It's the key exception to the general rule.
Good to know. Now, we talked concepts, but where's the hard evidence for the general dose rate effect?
Oh, tons of it. Decades of in vitro experiments. Classic studies by people like Eric Hall, using cell lines like HeLa, Chinese hamster ovary cells. They all clearly show survival increasing as dose rate decreases.
So multiple labs, multiple cell types confirming this.
Yep. You can plot composite survival curves for various human cell lines, and the trend is clear. Lower dose rate generally means higher survival, though you do see variability reflecting differences in repair capacity between cell lines.
What about in actual living systems, in vivo?
Strong evidence there, too. A really well-established model is the jejunal crypt cell survival assay in mice. Those gut lining cells turn over rapidly.
And they show a dose rate effect.
A dramatic one. For instance, studies show you might need, say, double the total dose delivered at a very low rate, maybe 5 milligray per minute, to get the same level of crypt cell killing as you would with a dose delivered acutely at like 2 or 3 grays per minute.
That's a huge difference.
It really highlights how much repair must be happening during that slow, low dose rate exposure, and possibly repopulation too, given the extended time frame of the low dose rate delivery in those experiments.
Okay, let's try to make this stick. Mnemonics can help sometimes. Any ideas for the general dose rate effect?
How about RATE?
RATE.
Okay, tell me more.
It stands for Repair Allows Time for Escape.
Repair allows time for escape. I like it.
Think of the cells escaping lethal damage because the repair systems have enough time to work. A slower dose rate gives repair more time, letting more cells escape being killed.
Simple connects the concepts. RATE. Good one. All right, let's pull out some key takeaways. Clinical pearls, if you will. Things to really remember.
Okay, hit me. Pearl number 1.
The big one. For low LET radiation, lower dose rate generally means more repair, less cell killing, higher survival. That's the default rule.
Pearl 2.
Remember the exception, the inverse dose rate effect. Specific low rates, specific cells, more killing. Mechanism. G2M accumulation. Curl 3. The dose rate effect isn't linear. It's most pronounced in that intermediate range, roughly 0.01 to 1 gray per minute. Outside that range, the impact flattens out.
Good point. And Pearl 4.
This is crucial. Low LETVS, high LET, low LET radiation like X-rays shows a big dose rate effect. High LET radiation, alpha particles, neutrons.
It's really damaging stuff.
Yeah, the damage they cause is dense, complex, much harder to fix. So high LET radiation shows little or no dose rate effect. The speed of delivery just doesn't change the outcome much because the damage is so severe anyway.
That's a critical distinction.
Yeah.
Okay, let's try a mini case. Think clinically. Total body radiation, TBI, before a bone marrow transplant. We often use a low dose rate spread over days. Why not just blast it quickly?
Great example. That protracted low dose rate TBI is a direct application of the dose rate effect. We're deliberately exploiting it.
How so?
We want to wipe out the marrow and leukemia cells, yes, but we desperately need to spare critical normal tissues, especially late responding ones like the lungs, which are often dose limiting.
And those tissues have good repair capacity.
Generally, yes. Better than many tumor cells, potentially. By delivering the dose slowly at a low rate, we give those healthy lung cells and other normal tissues maximum opportunity to repair the sublethal damage accumulating over the hours and days of treatment.
While the leukemia cells maybe don't repair as well or cycle differently.
Exactly. We're trying to widen that therapeutic window, kill the target cells while minimizing the damage to the dose-limiting normal tissues. The low dose rate helps achieve that differential effect.
Makes perfect sense. So, understanding dose rate isn't just academic theory, it's core to clinical decision-making.
Absolutely fundamental. Brachytherapy is probably the most obvious place we see it play out daily.
Right. Low dose rate brachytherapy, Think prostate seeds, I-125, or maybe older GYN-CS137 implants. They deliver dose continuously over days, weeks, even permanently for seeds.
That very low continuous dose rate allows for significant repair in the surrounding normal tissues, which is key to tolerance. The tumor gets a high total dose, but the dose rate effect helps spare the bladder, rectum, etc.
Then you've got the flip side. High dose rate brachytherapy, HDR.
Yep. Super powerful source, treatment delivered in minutes per fraction.
So almost no time for repair during the fraction.
Exactly. The biological effect per gray is much higher with HDR than LDR. That means you need meticulous planning, incredibly conformal dose distributions, because you don't have that ongoing repair helping to protect normal tissues during the delivery itself. Fractionation becomes key there.
We just talked about low dose rate TBI for transplants, another clear example.
And think beyond cancer treatment. Assessing risks from occupational exposure or environmental radiation like radon. These are typically chronic, very low dose rate scenarios. Dose rate effects are critical for estimating the actual biological risk, which is much lower per unit dose than for an acute exposure.
And radiation accidents too, right? The dose rate during the exposure would heavily influence the severity of acute effects.
Absolutely. Now, that inverse dose rate effect we discussed.
The G2 block thing.
Yeah. Fascinating biology, important for exams, but direct clinical application. Pretty limited.
Why is that? Seems like you could exploit it.
It's just too hard to precisely control the dose rate within that very narrow therapeutic window inside a tumor volume while avoiding spilling that sensitizing dose rate into surrounding normal tissues. The geometry and physics are challenging.
So more of a biological curiosity with limited practical use for that specific effect.
Right. But the general principle that lowering the dose rate spares normal tissues via repair that underpins all of LDR brachytherapy and influences things like TBI fractionation, that's clinically vital.
So the main treatment techniques where these dose rate principles really shine are
Brachytherapy, definitely. Both LDR, like permanent I-125 seeds, or temporary CS137.
where the dose is continuous over hours or days.
And HDR, where it's delivered rapidly in minutes over several fractions.
And that specific low dose rate TBI regimen for conditioning.
Those are prime examples where the choice of dose rate is a core part of the treatment design and rationale.
Okay, time for a board blitz. Let's see if we've absorbed this. Ready for some rapid fire questions?
Let's do it. Bring it on.
Question 1. Generally, for a given total dose of low LET radiation, decreasing the dose rate will likely A. Increase cell killing due to more direct DNA hits. B. Decrease cell killing due to enhanced tumor hypoxia. C. Increase cell killing due to inhibition of DNA repair pathways. D. Decrease cell killing due to increased repair of sublethal damage.
That's the main principle we started with. Lower rate, more repair time. So D, decreased cell killing due to increased repair of sublethal damage.
Correct. Question 2. The inverse dose rate effect observed in some cell lines, like HeLa S3, is primarily attributed to A. Enhanced DNA repair at very low dose rates. B. Increased production of protective heat shock proteins. C. Synchronization of cells in a radioresistant phase, like late S phase. D. Accumulation of cells in a radiosensitive phase, like G2M phase.
That's the exception, the counterintuitive one. It's about cells getting stuck in the sensitive phase, so deaccumulation in G2M.
Got it. Question 3. The dose rate effect on cell survival is most pronounced within which of the following dose rate ranges for low LET radiation? A. Less than 0.0001 Gemin.
Yeah.
B. 0.01 Gemin to 1 Gemin. C. 1 Gemin to 10 Gemin. D. Greater than 10 Gemin.
We called this the sweet spot, or the intermediate range where the balance is delicate. That would be B, 0.01 to 1 gray per minute.
Exactly. Last one. Question 4. Compared to X-rays, how would the biological effect of high LET radiation, such as alpha particles, typically be affected by changes in dose rate? A. A much larger dose rate effect would be observed. B. A similar dose rate effect would be observed. C. Little or no dose rate effect would be observed. D. An inverse dose rate effect is always observed.
High LET means complex, hard to repair damage. Speed doesn't matter as much, so C, little or no dose rate effect.
Perfect. Nice work. Okay, let's quickly summarize the key points.
Sounds good. So we covered the general rule. For low LET, slower delivery equals more repair, higher survival. Shapes the survival curve.
We hit the exception. Inverse dose rate effect. Lower rate, more killing sometimes, due to that G2M pileup in specific cells.
We identify that critical intermediate dose rate range where these effects really play out most strongly.
And stress the big difference. Low LET shows a dose rate effect. High LET really doesn't because the damage type is so different.
And finally, we connected all this back to clinical practice LDR and HDR brachy, low dose rate TBI, even thinking about chronic exposure risks.
Understanding dose rate, alongside the other 4 Rs of radiobiology, really gives us the tools to predict and maybe even manipulate how tissues respond to radiation.
Absolutely. For anyone studying for boards, these concepts, the general rule, the inverse exception, the LET difference, the relevant dose rate ranges, and the clinical links, they're definitely high-yield, testable material.
So we've talked a lot about repair during exposure. It makes you wonder, thinking about very, very low dose rates, like background radiation or occupational exposures over decades, does repair become so efficient that the traditional models based on acute effects even fully apply? Or do other mechanisms, like adaptation, start to play a bigger role at those ultra-low, ultra-protracted exposures?
That's a great question, and it pushes into really complex territory. It highlights that radiobiology isn't static. We're still refining how we extrapolate from lab conditions and clinical doses down to those very low-level chronic exposures. It's an ongoing area of research.
Definitely something to keep thinking about. Thanks for tuning in to this review session. You can find more resources and practice oral boards at radonsmartlearn.com and subscribe to Radon Smart Review for our next dive.